Hi everybody, I start second lecture. As you see here, I draw the genome of the COVID-19. It is around 30 kilobase and it starts from the 5 series here. It has two parts, which is important. It is for non-structural protein and structural protein. This genome, if you see, I draw the COVID-19. It is inside the envelope virus. And it binds to the M protein. M protein has two domains. And from both of domains bind to this positive sense RNA genome. Okay? As you see, for the non-structured protein, it has two parts, OLF, operating frame, 1A and 1B. These two segments help for transcription and translation of the non-structured proteins, which helps for the prophylaxis and prevention of the immune system effect, like proteolysis in the cell. Okay? And it has two parts, OLF1A and OLF1B. And the second segment is the structural protein part. We can produce SEMN protein, which can be seen in the structure. Okay? As you see, I draw here is M protein or nucleocapsid protein, and we have S protein or spike protein, which has the two parts, S1 and S2. And we have a E protein, envelope protein, and we have a membrane protein or matrix protein, which is the most abundant protein in the membrane. Now, briefly I explain what is the function of the genome and how we can think about the treatment and the prevention of this virus, and how we can save the life of the person when in the hard or severe situation. As you see here, in the ORF1A, and we see two parts which is very important. One of them is the papain-like protease. Okay? As you see here, these parts, okay, helps for the protein synthesis, which function is the proteolysis. Okay, and interferon antagonist. Mainly it prevents effect of interferon, okay, and also it helps for the production of the protein which has a proteolysis function. It is very important, as you see here, okay, as you see here, and it's very important. And the second segment is the 3CL protease. And it's also help for the synthesis of proteins or enzyme for the proteolysis. Then, mainly I can mention in the ORF1A, mainly protein synthesized in the transcription translation process, which has a proteolysis and immune system prevention properties. Okay, but in the ORF1B, we have three segments very important. The first one is the LDRP, or RNA-dependent RNA polymerase. As you see, as you know, for the replication, we need the RNA polymerase. Okay, and for this mechanism, we need the RNA-dependent RNA polymerase, which helps for the viral replication and transcription, because it's very important, this part. Then for this segment, okay, this function is the RNA replication and transcription, also help for the production of the mRNA RNA. It's very important. And the second segment is the helicase. Helicase, as you know, it can open the DNA strands, and is important. And if we want to do transcription, we need a helicase to open the double DNA strand, because we can copy the genome from the DNA. It's very important. And the last one, endoribonuclease, as you see here, as helicon open the DNA helix. And endoribonuclease also help for the RNA replication. Okay, and the last step, it is until, as you see in the ORF operating frame 1B, mainly RNA replication and transcription is important. We have to mention it. And the last part is the structure protein, which helps the production of the spike protein, envelope protein, membrane or matrix protein, nucleocapsid proteins, which helps for the mechanism for the production of the protein. Now we go to the mechanism which what is the function of these proteins? Okay, the first protein is M protein. As you see, M protein has a two domain, and both of them, these both domain, bind the RNA genome from the both two sides. Okay. In this situation, if you see here, both domain bind to the RNA. Okay, and it's very important. And when this excuse me, and when it bind, it helps for the DNA replication. Okay, genome replication. And also helps for the transcription. It's very important. Okay, the second protein is the protein or envelope protein. Envelope protein play a role in the viral budding. For example, when the virus COVID-19 synthesized inside the cell, now viral needs to budding goes out from the cell. Okay, and for the viral budding and viral envelope morphogenesis, we need the proteins, very important. Then it's very important. I have to mention about this, and it helps for the increase the speed of proliferation and budding of the virus from the cell. It's very important. Okay, if you see, it is on the membrane, and also it helps for the membrane fusion. And the second protein, also third protein, excuse me, protein is the most abundant protein on the envelope membrane, and we say it's membrane or matrix protein, and it is responsible for the shape of envelope. It's very important, and it also helps for the viral fusion and the assembly, and also it has a connection with the genome. Okay, it's very important. I mention here there's a relation between the protein and the genome. Usually, mainly when we have a resistance or mutation, we need a mutation about virus. It helps. It's very important, and the other protein is the S protein. M protein I speak about, and protein, and the second last one is S protein. S protein has two parts, S1 and S2, and these two protein helps for entering the cell. These proteins can bind to the ACA2 receptor. It's very important, and we will see in the lecture how this mechanism happen, because the S1 subunit bind to the host cell. S1 helps to the binding to the host cell, and important, and S2 subunits help for the fusion of the viral genome and the cellular membrane, and the fusion of the viral and the cellular membrane. It's very important for this section, and in the next lecture we will study mainly about the pharmacological effect and how we can use a treatment, how we can prevent the virus proliferation. It's very important. I hope it will be good, and thank you for paying attention. I hope see you in next lecture.